Well, so I think we can start.
Yes. Hi to all. We are from Ukraine. Here in the bottom of the slide, our emails. You can use them if you want to get this presentation or the reference list from our speech. Please be free to write to us. Next slide, please. So, since the days of the dinosaurs, everyone has wanted to know how to properly analyze the results of a single cell RNA sequencing in order to accurately model the processes of cell differentiation. But statistical mechanics appeared only in 19th century. Minds smarter than dinosaurs have come up with Fokker-Planck equations, Spearman correlation, Shannon equation, and Dirichlet distribution, and then a group of people suddenly decided to use them for the benefit of biology. And we, Lera and Igor, are gonna tell you about what and how they did it. The cell type is determined by the genes that are transcribed in it. Single-cell RNA sequencing allows us to find out about the level of gene expression in a cell. The specialization of a cell can be imagined as its movement in state manifold, geometric representation of functional cellular states with the coordinates of gene expression and pseudotime, and changes in gene expression profile shows the cell's way. But no wet biologist would agree to prepare single-cell RNA sequencing every 5 minutes to keep track of the cells. Moreover, it's impossible. After all, the methods involves the destruction of cells. Therefore, we cannot know the exact previous state of the cell, thus inferring each cell's past and future lineage trajectories requires a model that probabilistically assigns representative ancestors and progeny of the cell of the given cell from the large ensemble of cells measured at earlier and later time points. Also, single cell dynamics is probabilistic, like probability motion. That's why statistical mechanics is the best approach to visualize cell trajectory. On slices of the cell path, we get not one position of all cells, but a set of coordinates, such called local attractors, which designate the cells that are next to the most stable state at this stage of the development. Assuming that the states are connected based on the laws of statistical mechanics, we build low energy passes, often called lineage trajectories. As biological developmental processes are fairly stable and generally do not depend on the specific series of steps that led to a given state, the cell trajectories between states are viewed as a memoryless mark of processes. This is how clearly you can visualize the cell development potency, and here is examples from several articles. And existing methods merely differ in mathematical approaches. So now we're gonna talk a little about different methods. Uh, Deschendorf and Feinberg, in their recent article, divided the methods for visualizing the cell potency into top-down and bottom-up. By developing of methods, the authors of those tried to bypass such obstacles as lack of knowledge of tissue marked genes, where differentiation hierarchy are not well established, overfitting, and smooth out noise. Bottom up assumes potential function, which given some input of only one cell and outputs an approximate value for the cell's differentiation potency. Top down begins by analyzing all the cells to infer differentiation potency of each cell. So let's go in detail. Top down methods. PBA and Waddington optimal transport. PPA is an algorithm which uses population balance equation to infer cell state dynamics from static snapshots. This equation states that the rate of change in the number of cells equals the net cell flux into and out of the region of state manifold. Still, there is a problem. Multiple dynamic trajectories can generate the same high-dimensional population snapshots. That's why we need to construct our model by assuming there's no hidden variables and there's no oscillations in our system of cells. Due to this assumption, we can use Fokker-Planck equation. The output of PBA is a discrete probabilistic process that is a Markov chain that describes the transition probabilities between the states. And on the base of transition probabilities, we can compute fate probabilities and temporal ordering of cell states.
Well, and now I'll tell a little about Waddington optimal transport method. So it's also like a top-down method, and it uses optimal transport theory for reconstructing probabilistic cell state trajectories. And in this method, with the help of optimal transport, we can calculate so-called time couplings. What is that? So it's a joint distribution between consecutive time points. So the authors of the article you can see here, they call it transport maps. So, and then when we infer due to solving of this optimal transport problem, due to these transport maps, we can infer couplings over long time intervals simply by composing transport maps between every pair of consecutive intermediate time points. And in this model, we also assume that cell differentiation is a Markov process. And moreover, in the Waddington OT, we capture only time varying components of the distribution. Well, trajectories are defined in terms ancestor and descendant distributions. And so how can we infer them? So descendant distribution we can infer by multiplying the vector of cells at some time point ti. And so to infer the descendant distribution at some point ti plus 1, yeah, so we multiply the vector of cells in time ti by this transport map. And to infer the ancestor distribution in time ti minus 1, we multiply the transport map by the vector of cells, so by just matrix multiplication. So and you can see in the picture here the great, in my opinion, visualization of these cell trajectories in time. And I just want to show you the video of the evolution of these cell fate trajectories. Just a moment, I just need to. I think you can see this. Yes? Okay. So here is the evolution of cell differentiation over time. So we can see this different clusters of differentiated cells here. Yeah. And so we can make a transition to bottom-up methods. And again, Valeria will tell you about them. Just a moment. I need to turn off my mic.
So that video that Igor showed, I think it's amazing. The result of applying those methods. Uh, Now we turn up to bottom-up methods, and there should be a little pre-story about them. The pluripotent cell does not express preference for any particular lineage. Please keep that in mind. A cell's choice to commit to a particular lineage is a probabilistic process and can be characterized by the state of high uncertainty um, or entropy, because all lineages choices are equally likely. Entropy is a generic term to quantify the amount of uncertainty associated with the system or the outcome of some measurement. It could be viewed from a perspective of cellular plasticity as probabilistic change in lineage specifying transcription factors from pluripotent cell to high differentiated. Also, it correlates high openness of chromatin and relatively the amount of mRNA synthesized. The first method is STENT, the terms for single cell entropy. So STENT quantifies the degree of uncertainty of a cell's gene expression levels in the context of a protein-protein interaction network. In a sample with high expression of gene 1 and gene 2, the two corresponding proteins are more likely to interact than in a sample with low expression of first or and second gene. That is the assumption that leans in the basis of STENT. And signaling entropy can accurately discriminate pluripotent from multipotent and differentiated cells. A key feature of STENT is the assignment of each cell to a unique potency state and co-expression
I'm sorry,
cluster.
Valeria.
Yeah.
Think we are just on time. I don't know if you want to jump to conclusions, if you are almost at the end, but we are just finishing the time. I'm sorry.
So sad. A lot of information is missed. Okay, let us tell a little bit generally about the third category of methods. And on that point, we'll stop. So there is another hidden method, hybrid methods, and they borrow elements of a bottom-up approach that require data from other cells to estimate the potency of any given cell, which we further refer to as hybrid methods. And there are two of them exist now, single-cell entropy and trace. Then we will stop on that. If you're interested in that topic, please contact us. There list of references at the end. And thank you for your attention.
Thank you to both of you. I'm sorry that I had to cut you. I thought that you had a little bit more to talk about.
That's also our fault. We had to manage the size of the speech.
Yeah, it wasn't